Okay, let's go. Welcome to my presentation on structural colour. We live in a very colourful world, and most of this colour comes from two different processes: pigment colour and structural colour. In pigment colour, a sample contains a special chemical called a pigment, which reflects only the wavelength of light of the colour of the sample. Now, that sounds a bit complicated, but effectively, if you have a sample, for example a leaf, it contains a chemical such as chlorophyll, which absorbs all wavelengths of visible light except for green, which it reflects, and therefore we have green leaves. The other type is structural colour. Now, structural colour is different because it doesn't use any kind of chemicals, and its colour comes solely from the microscopic structure of cells or crystals or feathers, and as a consequence, it gives very brilliant, iridescent colours. So let's have a comparison between the two. On the left-hand side here, we have a red card, which is coloured by a red pigment, and you'll see that as we change the angle of incidence of light upon the surface, the wavelength of light doesn't really change, it stays red. And that's because the pigment doesn't depend, the colour coming from the pigment doesn't depend on the angle of the light incident on it. But if you look at here, a sample of blue morpho butterfly, as we change the angle of the light incident upon it, we get very bright flashes of blue, and the wavelength of the light coming off of it changes. And this is because the structure is dependent on the angle of incident onto the sample, and this is what my experiment is all about. So what are the aims of my experiment? Well, we hope to gather an understanding of how small-scale structures can produce such brilliant colours, and we hope to create theoretical and experimental models and compare the two and see if they differ in any way or how they're similar, and we hope to identify any errors involved in this to make sure that we're good scientists. So let's have a closer look at the beetle structure, shall we? So on a very, very small scale, the beetle is effectively made up of layers of translucent materials with different refractive indices. In this case, I've only done 4, but in actual cases, one that we look at later, we can get up to 19 and even more. And as our wavelengths of light come in upon our sample, some of the light is reflected off, but some of the light refracts through and goes inside of the material. And on this new boundary, it reflects again, and refracts, and reflects, and refracts, all the way through the sample. And effectively, you have a big sort of array of light coming off of the sample, but all with slightly different path lengths due to this refraction and the distance between the samples. And some of this light will constructively interfere, so here and here the troughs are lined up quite nicely, but here and here they are opposed, and so you would expect to see destructive interference. As our angle of incidence changes, our path length will also change, which will give different colours, which is why the angle of incidence gives a different colour depending on what angle you look at it. So let's have a look at the experiment that we set up. So this is our first setup. This is to look at a FBH63010 Thor Labs optical filter. A lot going on there. But that's the filter there, this little red thing here. And this is the experiment setup that we had. So we have a halogen light producing nice white light that went down an optical fiber into a collimator. This allows the light to stay true on its path through our sample. And when it gets to the detector on the side, we pass the light into a spectrometer to see the makeup of the light and into a computer so that we can observe it. This is the software that we use. This software is called OceanView. So we were hoping to look onto the internet and find images or descriptions of the structure of the filter, but we couldn't find anything, and I think that it is a bit of a trade secret. So instead what we did is we looked at the schematics, and we looked at the description of the filter, and we found that it had to have, for light travelling straight through the filter, a peak at 630 nanometers, a bandwidth of 10 nanometers, and we expected that the wavelength would drop as the angle increased. This is the data that we gathered. So this is the heat map that we created. On the x-axis we have the angle of incidence, on the y-axis we have the wavelength, and the intensity is represented by the colour here. Our peak wavelength at 0 degrees is 630 nanometers. Tick, very happy. Our bandwidth is about 10 nanometers, so very happy, tick. And we can see here that our peak wavelength drops as our angle increases. So we're all very happy. You can see an image here of, oh, it's not there. It was doing it earlier. But very happy with all of this. Tick, tick, tick. Good bit of data collection. The second experiment we did involved reflection. So we used, this is the setup. It's a little bit more complicated, but it effectively is all, most of this gadgetry is about making sure that the light travels the right path, is not polarized in any way, and hits the sample in all the right ways. But dumbed down, this is what the setup was effectively. We had a halogen light source come through an optical fiber into our emitter, which is this thing here. And it would travel down these arms and strike a sample, come off of the arms, go through a spectrometer, into a computer, very similar as before. But this time we would change the angle of incidence and the angle of the detector as we collected our data. And once again, we used trusty OceanView as our spectrometer software. Right, so what did we expect to see? So this is our sample. If the video starts playing, it should shake and load. Yep, that's perfect. And so as you can see, this is very shiny, very green. And so that was the key thing. It had to be of green wavelength, which is about 570 nanometers. We went and looked at some theoretical models seen in papers. I'll reference them at the end. But this is one that we found, which was a zoomed-in version of the structure. And each of these layers are different materials that the light shone through. We had the refractive index, we had the pixels, so we calculated the lengths. And once we had these lengths, we put them into a online structural color software calculator thing, and it gave us this at 10 degrees, and we extended that all the way out to 45 here, as we did the same in our actual experiment. And this is the theoretical heat map of the C. Raja beetle that we gathered. Let's see what we actually got when it came to experiment. This is what we actually gathered. Very different. Lots going on here. For starters, the big glaring issue that we found is that our theoretical model was a lot lower wavelength-wise than our experimental data. On top of that, our experimental model continues, all the way through to 45 degrees without dropping off in intensity. For us, it didn't do that at all. It completely dropped out. And there are also these extra bands that appear. Now, we're fairly confident we know the reason why this is. This is for a very idealized version. There isn't any absorption. The sample that we looked at is specifically just one part of the sample, but this can be for any part of the beetle that we were looking at. So there's just lots of biological noise, you could say. So we tried to cancel that out by adding in absorption, which is our new here it is. Now a bit better. Our wavelength has gone up closer towards our theoretical, not our experimental model, but still not the same. And our intensity does drop off a bit. It's a bit lighter there, darker there, but still not the perfect one. And we are still sure, we're still convinced that this is because of general sort of biological differences between a perfect theoretical model and real life. But what are the errors associated with an experiment like this? So there are lots of things that can go wrong, mainly as you saw the fact that biology and physics are quite different sciences. And on top of that, when moving the detectors, you can get different changes in intensity, but very slight changes in angle, and we had to sort of analyze which ones were best for us. But the way that we've sort of analyzed our data is by taking measurements of the same sample weeks apart, and we thought that if anything changes significantly in them, we did something wrong in one and there was a systematic error. And I'm quite happy to say that 2 weeks apart, we've got very similar data. Now, the second peak here is slightly harder than the second peak here, but they're still there. The wavelengths are the same, the intensities drop off the same, so super happy with this data. It's safe to say that we're not making any serious systematic errors. But what's next for our experiment? Earlier in this presentation, I lied a little bit about structural colour. I showed you a blue morpho, my favourite example of structural colour, and I explained how it works like this. In actual fact, it's much more complicated. There's very tiny, intricate details that are part of the butterfly, which are not just simple layers of different materials. And our hope is that moving forwards in this experiment is that we'll model this in the same way that we modelled our C. Raja beetle. And what we hope to do is we take layers of the sample, and we use the refractive indices of each of these layers and then times them as a percentage of the layer. And so we hope to get many, many layers. Obviously this is just 4 and there's a lot more here, but we've got many layers of different refractive indices with different lengths going down, and we hope to compare that to any experimental data that we gather for the blue morpho butterfly. But yeah, so that's pretty much my experiment. That was my presentation. Thank you very much for watching. If you have any questions, email me, I suppose. These are some of my references, but thank you very much. Let's go, Tom! Ow!
Oh shit, you're right.
Yeah, that's sick, man.
Fuck it out. Absolutely bossed it. Ideal. Woo!
They're both uncharged.
No, you're
I'm kidding.